Hey guys, Miss Cote here, and welcome to the first video in the Gene Expression and Regulation unit. This video is all about DNA, the molecule that holds all of our genetic information. You may remember from honors biology that Watson and Crick worked out the structure for DNA back in 1953 and ended up winning a Nobel Prize for that discovery. In this video, we're going to look at the history of DNA and how it was accepted as the genetic material of the cell. So let's get started. In the last chapter, we saw that scientists were expanding on Mendel's work, and the chromosomal theory of inheritance was put forth around 1902-1903. We also looked at two major contributions of Thomas Hunt Morgan and his work with fruit flies, providing evidence that genes were located on chromosomes. Knowing that the two components of chromosomes were DNA and proteins, now the search was on to figure out which one of these two biological molecules was actually the genetic material. The first experiments to suggest that DNA is the genetic material was done by Frederick Griffith in 1928. Griffith used two strains of a bacterium that causes pneumonia. One strain was pathogenic, disease-causing, and the other was totally harmless. As a positive control, he took the pathogenic bacteria and injected it into the mice, and the mice died. As a negative control, he took the non-pathogenic bacteria and injected it into the mice, and the mice were fine. Then he took the pathogenic bacteria, heat killed them, and injected them into mice. The mice lived. However, when he mixed those heat-killed bacteria with the live non-pathogenic bacteria, the mice died. Upon further investigation, he found that some of the living cells had now become pathogenic to kill the mice. He used the term transformation to describe what happened. A cell took up external DNA, and that DNA was able to change the cell. This work was continued by Avery, McCarty, and McLeod in the 1940s, and the transforming substance was found to be DNA. However, many other scientists still had their doubts. The next round of experiments were done in 1952 by Hershey and Chase using bacteriophages, or just phages. These are viruses that infect bacteria. They designed a simple but effective experiment, taking advantage of the fact that these viruses are only made up of protein and DNA. In the first batch of experiments, they used radioactive sulfur to label the proteins of the phages. They allowed the phages to infect bacteria, and then they threw everything in a blender to free the protein coats of the viruses from the bacteria cells and spun the parts down, knowing the bacteria cells would form the pellet and the viruses would be in the supernatant. At the end of it all, the radioactivity was found in the liquid, so the proteins did not get into the bacteria cells. In the second batch of experiments, they used radioactive phosphorus to label the DNA of the phages. They followed the same procedure and found that the radioactivity was found in the pellet, so the DNA was able to get into the bacteria cells. So they concluded that the genetic material was the DNA. Around the same time, Erwin Chargaff was doing his work, focusing on the nucleotide composition. It was known that DNA was made up of nucleotides, and each nucleotide had a nitrogenous base, a sugar, and a phosphate group. Chargaff was studying the DNA from various organisms, specifically looking at the composition of the bases, the percentage of A's, T's, C's, and G's in an organism's DNA. And while he found that different species have different base compositions, he did notice a pattern. The percentage of A's always equaled the percentage of T's, and the percentage of C's always equaled the percentage of G's. These two findings became known as Chargaff's rules. We usually refer to the second one as the base pairing rules, which we will get to shortly. Now that it was widely accepted that DNA is the genetic material, the next step was to figure out the structure of DNA. Rosalind Franklin was a chemist who was an expert in X-ray crystallography, a technique used to visualize molecular structure. She and her assistant, Maurice Wilkins, were the first to photograph DNA. This is a famous picture called Photo 51. It was this picture that helped Watson and Crick not only figure out that DNA was a double helix, but to also work out the dimensions as well. Based on the photo, it was concluded that there were 2 sugar phosphate backbones, and the nitrogenous bases from each strand pair up in the center. Watson also worked out that the 2 sugar phosphate backbones run anti-parallel or opposite to each other. From top to bottom, the strand on the left runs 5' to 3', and the strand on the right runs 3' to 5'. The direction of the strands will become important when we start discussing DNA replication, which will be in the next video. Another puzzle to solve was how the bases pair up. At first they thought that like paired with like, so A pairs with A, G pairs with G, and so on, but that did not produce the uniform width shown in the X-ray. Pairing 2 purines together made the helix too wide, while 2 pyrimidines together made the helix too narrow. Watson worked out that to make the helix uniform, a purine must always pair up with a pyrimidine. And going back to the Chargaff rule that the percentage of A's equaled the percentage of T's and the percentage of C's equaled the percentage of G's, Watson and Crick determined that this must be how the bases pair up. These base pairing rules also aligned with the purine and pyrimidine pairing, which keeps the helix a uniform width. And looking closer at these base pairings, it also works out that the way these bases are built, there are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between guanine and cytosine. And please don't think you need to memorize these structures, because you don't. So that's it for the history of how DNA came to be recognized as the molecule of genetic material. The next video will look at how this structure of DNA lends itself to the accurate replication of the DNA molecule, which is vital to the multiplication of our cells. So until next time, be a good human.